Sánchez is an ICREA professor at the Institute for Bioengineering of Catalonia, where he leads the Smart Nanobiodevices group, which develops different systems ranging from active nanoparticles, nanobots, 3D bioprinted actuators, and flexible biosensors to be used for future nanomedicine and environmental applications. He's also Deputy Director for the Internationalization of IBEC, an honorary visiting professor at Harbin Institute of Technology in Shenzhen, China. Before that, he worked at the Max Planck Institute for Intelligent Systems in Stuttgart and at the Institute for Integrative Nanosciences in Dresden, at Mana NIMS in Japan, and at Technical University of Twente. He received the ERC Starting Grant in 2013 and the ERC Consolidator Grant in 2019, and 2 ERC Proof of Concepts. He has received several awards and recognitions, such as the Princess of Girona Scientific Research Award in 2015 and the National Research Award for Young Talent in 2016 by the Catalan Research Foundation. So now, Samuel, thank you for being here today, and the floor is yours.
Thank you, Teresa. Thanks for the introduction, and thanks for organizing this online seminar. I want also to extend my gratitude to everyone who is connecting from all parts of the world to listen to this seminar. So the work I will be presenting today is basically the work from all these very motivated guys who are taking part in my group at the IBEC. And today I will tell you 2 topics. Both are based on bioengineering hybrid devices or hybrid machines, and I will go from nanobots, which are nanosystems, all the way to the 3D printed biobots. Probably I will run not into many details. I will give an overview of a few of these things, and probably it will take too long. Sorry for that, but I hope you enjoy the talk today. And let me start with the motivation, and this is inspired by 2 very visionary books by Isaac Asimov. And the one on the left side is the first part of my talk. It's Fantastic Voyage, how to make tiny submarines that will go through the body and heal and cure some human beings. And the one on the right side is how to create robots, right? So at a larger scale. So on the first part, I hope I will convince you that we can do at least some parts of these tiny submarines, some things that move in fluid in a controlled manner. And I will focus today only on a particular point of biomedical application. So going across all these length scales, we are able to replicate hybrid systems. Hybrid, what you'll hear now, is about combination of biological and artificial parts, and try to mimic what we find in nature. So on here we have mimicking ideas of how to make bacteria or virus. Also, we want them to move collectively, like the swarm fish, and all the way will be like making tissues embedded into an hydrogel to make muscles that actuate. So all these are hybrids, and I will tell you now why. So, but first I will focus on how to make nano systems or nano swimmers like bacteria and virus. So again, we look always into nature, and we are inspired by the shape, structure, and some of the functionalities. And looking at the nature, we can see cells, bacteria, and viruses, which are like structurally are simple in a way because they are like spheres. They also can have this hinge here, or they are elongated rods. And that's what we copy in our lab. We make small particles about the size of 2 to 5 micrometers in diameter. We can make also nanotubes on the size of a couple of hundred of nanometers and a few nanometers in length. And we can make also virus-like structures from 50 nanometers, 100 nanometers, and so on, and very rough surfaces. So all this is trying to mimic what we find in nature. But to give you also like this is kind of a teaser, right? So what you're gonna see now, I'm convinced that we are actually mimicking it so much that I'm kind of confusing you here. So if I will ask you who thinks the one on the left side is biological, or who thinks the one on the right side is biological, probably will never finish this talk because now I don't know who is raising your hands, but just think about that, right? So how similar the one on the right and is to the one on the left. But in fact, this one, I don't know if I see here. Okay, so here on the one on the right side, what you are seeing is the smallest jet engine in the world, which is fully artificial, is made by us, by Shima and myself when we were in the Maximum in Stuttgart. So we had a nanowire, nanorod, functionalized with catalase, with urease, and we put in solution and they start to swim, okay? So this, if you see again the videos, now these on the left side, these are bacteria which are swimming near surfaces. So I hope with this video, we're not gonna end up the talk, of course, but I can convince you that we can replicate some of the features of bacteria, like swimming in this case, okay? So how do we get to this point? So this will be the final part of my talk, but now how we get there? And what for, by the way? So why we want to do that? And if we go back in time again, so we can take the book and the talk from Richard Feynman, where there's plenty of on the bottom, the talk he gave at Caltech, and he gave some hints on how to create the nanomachines, but even more, he actually gave credit to his student Gibbs, where he said, imagine that one day we could swallow the doctor, similar to the book, which was, by the way, afterwards in Fantastic Voyage. So can we make a small submarine, a small nanoparticle, and go and swim inside your body and do something like a doctor will do externally? That was the question. So nowadays is what we call precision medicine or nanomedicine. We want to make some tiny devices that will go inside and then get to the center of a tumor. But despite the recent advances in the field of nanomedicine, this is probably around the last 20 years, the lipids, polymer nanocarriers, inorganic viral carriers, and drug conjugates. Report says that only 0.7 of the nanoparticles designed to treat tumor reach to their target. So as you can imagine, the efficiency is very, very low, and we want to find ways on how to improve it. By now, like passive transport, or what we call in nanomedicine, these nanoparticles that I just showed you in the previous slide are based on this passive transport, which means you put in the bloodstream, you put into the body, and they don't have any control. They go everywhere, and you see probably it's very, very rare that they touch the edges, where actually it's very important to reach. But you might agree that if we make an active transport, like a boat, something that can pack the cargo inside, can go from point A to point B efficiently, this will be much better in terms of these 0.7% efficiency numbers. But swimming at the nanoscale is extremely challenging. Purcell reported in this seminal paper in 1977 what is called Reynolds numbers. This is the ratio between inertia and viscosity. So if we put some numbers, a human being swimming in the sea, so we'll have a Reynolds numbers of 10 to the 4. It means that the inertia is extremely high compared with this viscosity. But as we go down in size, the viscosity is predominant and the inertia plays no role whatsoever. So this is very difficult to overcome. And he also referred to imagine that you are swimming in a pool full of honey. So how viscous this will be. As soon as you stop swimming, stop paddling, you don't go anywhere. So making something tiny is complicated. Making something tiny that can swim is extremely complicated. But you might say, so how does nature achieve this movement? Because we see bacteria moving, right? So this, one of the ideas, or one of the ways to achieve this motion is through catalysis, converting chemistry, all the chemistry which is around it, like the chemical fuel that we call it in our field, by chemical reactions into motion. So that is what we do. Okay, so we have a nanoparticle which has an engine, has something that takes the fuel from the surrounding and converts it into motion through chemistry. Okay, so which are the minimal components of these nanosystems, of these nanorobotic systems? So there are two components. The first one will be the chassis. So we need something to anchor, like a vehicle, right? Well, like a car, we need the chassis. Or in our group, we have developed many different structures, like nanoparticles, core shell, hollow particles, tubes, and mesoporous tubes, and also hollow capsules here, or Janus particles, and so forth. And for a while, we have been working with mesoporous silicon, mesoporous silica is a FDA-approved material. It's highly porous. You can easily functionalize. You can easily change the charges. You can easily change the shape and size. Sorry. So it's very beneficial for us. Say that it's also true that we are currently using other type of materials to explore different functionalities, but this is so far a very good chassis for us. Second component will be the engine. So we need a functional component to provide this motion. In the field of nanomotor, platinum has been used for catalytic reactions, titanium dioxide for photocatalytic, gold for light activation, and enzymes for biocatalysis. We're very interested in enzymes because it gives biocompatibility. So imagine you have an enzyme. So actually we have 1,300 enzymes in a human cell and 75,000 enzymes in our body. So each enzyme is specific for at least one substrate. So you can imagine you put these nanoparticles coated with the enzymes inside our body, and they will catalyze a particular fuel which is in a particular location. And this reaction will lead to a flow around and will move the particle forward. I will get to that point later, but this is it makes the system fully biocompatible. You don't need to add the fuel externally. It should find it available there. Not only that, but if we look in the literature, we can see that enzymes feel chemotaxis. We can have cascade of enzymes, like the substrate of A will lead to the product, which is in turn the subset of enzyme B and C and so forth, and that leads to a communication and cascade reaction that moves them collectively. This was reported by Sen. Also, you can anchor a cascade of reactions inside a polymersome, and this leads to chemotaxis. This is also very interesting that this is an helix which is moved by magnetic field by Per Fischer and co-workers. And on the top, they have similar to Helicobacter pylori, they have urease. Urease will change the pH, and actually what we'll do is change the viscosity, so they will move further. And also Henry has report column flux dynamics. So these enzymatic reactions are very, very interesting overall. So what I will be presenting today, I will tell you about the questions. Right? We have questions in our mind and we want to explore them. So which are the fundamental aspects, and then also towards, once we understand them, we can go towards biomedical application. So the first question will be, which are the best enzymes for propulsion? How many enzymes we need? Or what's the distribution around, and what determines the dynamics? So what we did in our lab is to take 4 different enzymes which have completely different K-cut, so catalytic turnover rate or efficiency. So we take aldolase, it's very slow, glucose oxidase, acetylcholinesterase, and urease. We have a mesoporous microparticle, and then we coat the ring, so around the particles with these enzymes, and we observe the motion. Okay, so here you can see the tracking trajectories for the different enzymes, and here we plot the speed versus the K-cut. So just glancing over the figure, you can see that the faster the enzyme, right, which will be the urease, the faster is the speed of the micromotor. But it's not only that, and we also wanted to explore more complex and structural dynamics, or what is at the fundamental level. So let me go back here. So this is the typical plot we have. Is the mean square displacement means the displacement of the area explored by the micromotor when we add the fuel. So here, for the higher the urea, this is for urease, the higher the area explored. For acetylcholinesterase, it moves, but not that much. But then if we take glucose oxidase or aldolase, there's seldom movement. So there's almost no movement at all. So we collaborated with Silvia Osuna's lab, and they did the molecular dynamic simulations, and they can see also some structural features like the conformational changes, the flexibility of the enzyme is also crucial for the motion of the particles. So this opens new ideas on what should we care about when we select an enzyme. Okay, so also the flexibility. So the most flexible enzymes for us was the one giving higher propulsion. In this case was the urease and acetylcholinesterase. Okay, so for microparticles to move, we need an asymmetry. We need the engine to be on one side. And this is a little bit of state of the art, like these drawings. And basically, most of the particles in the literature are based on Janus structure. Janus is the god, the Greek god of the two faces, where the chemistry or the reaction in this particle is in one of the two phases, okay? Can be here, it can be inside and outside, right or left, and so on, either for nanoparticle, for microparticle. But our question was, what if we take a particle and we fully coat it, right? We can remove all the fabrication steps and then make a particle move. Will it move or not? So in principle, if it's fully coated and all happens at the same time, there's no asymmetry, it shouldn't move, but it will simplify the fabrication, reduce the cost, increase the yield, and will be fully biocompatible because we don't need anything else there. Going back in the history of motors, just to give you some idea, the Janus particle was basically coated with platinum, half coated, and this will create a chemical gradient around the particle and will push the particle forward, as you can see here. Always the platinum is on the backside. Okay, but now what we do is we don't have half particle. We take the urease, we coat our particle, and you see we don't predetermine any coating. We just put it there and we see what happens. And what happens is the following: they do move. Okay. So even though it has said again that we need this 50% coating or a Janus structure, coating our particles without any asymmetry, it moves. So how can we prove whether there's asymmetry or not? So we collaborated with Lorenzo Albertazzi's group, who actually gave the seminar last week, and we used their storm. And here actually, what we did is we label, so these are microparticles labeled with enzymes. This is imaged by fluorescent imaging microscope. So as you can see, a priori, you cannot predict any asymmetry. But if we run the super resolution microscopy, which is what is done here, this is stochastic reconstruction, by blinking the dye, we can reconstruct and we can see already some gaps. Okay, so these are 2D projections, so you can see already some asymmetry. And even more, if you run the 3D structure, you can identify every single dot as every single enzyme on the particle. Okay, so then we run some code to check the density map, as you can see here, and then the blue dots are the high dense patches. And in every single particle we investigated, we've seen patchy-like structure. We cannot a priori say how many patches, we can a priori not say what will be the density of enzymes, but all have this local asymmetry, which is enough for leading to a motion of the particles, as you saw in the previous one. Okay, so not only asymmetry was important, as we reported in this paper in JAX 2 years ago, but the number of enzymes is very important. So if we increase the concentration of enzyme, the speed is increasing. So this, what you are seeing is the tracking trajectories through a microscope. So the more urease we have in the solution, in the incubation solution, the more enzymes we localize. But to our surprise, there was not a linear increase on the speed. So it means that not the more enzymes you put, the linearly will increase like here, but there is a threshold. And that was very important because we didn't understand that. So that means there's a minimum amount of enzymes we need for the motion. And this can be also, confirmed by these 2D projection. So what do I mean here? Here we have a lot of enzymes. We see an asymmetry. Here's not too many enzymes, here there is enzymes, okay? So we see a tracking trajectory, so the particles do move. But if you reduce the enzyme number, you do have an asymmetry, but they don't move. So we need this crucial number here for the particles to move. Okay. So there will be also more things, but for the sake of time, I'm not going into detail what other parameters, other enzymes we're using, but I want to move now to shift gear a little bit towards biomedical applications. So what can these motors do now? Okay, so we have a multifunctional nanomotor. So we can draw load drugs, we can add linkers, we can add enzymes, we can add nanoparticles. So that means that we can use them for propulsion and actuation, but we can also use them for imaging and diagnostics. So again, I'm going to summarize here a few works that we have done. I told you in the beginning about like throwing things through the river or a boat. So we want to compare passive and active particles, and this is what we did here in this work. We took passive nanoparticles, mesoporous silica, and we had the mesoporous silica coated with enzymes, and we showed actually that there was a huge increase in the cell internalization into HeLa cancer cells when the nanoparticles are moving. Then we also use these nanomotors sensing by coating with DNA nanoswitches, so we can check whether there's a acidic pH or basic pH, and that was thanks to the collaboration with Francesco Rizzi's lab. And this was very important because cancer cells have a slightly acidic pH compared with healthy tumors, and this is what we actually exploited here. We coated the nanomotors with a gate, a pH gate that will only open once the nanoparticles are inside the cell. And today I will only focus on this one because I've been talking about the urea the whole time. And here what we did is the following. We took a 3D bladder spheroid, and then you will see in a second, okay? And then we explored the possibilities and the potential of nanoparticles. But why bladder cancer? So bladder cancer is one of the most common cancers worldwide, but I was surprised to know that it's the most expensive cancer to treat because all the diagnostic and treatments nowadays are not efficient enough. So that means that the relapse is extremely high, between 60 and 70%. Means that the ways they nowadays are identifying, so doctors identify the tumor and they treat it, are not the best so far. So we thought, why not to take our nanoparticles and see whether we can use it. And of course, there's a lot of urea inside here. So what we did is to have nanoparticles, silicon nanoparticles. We coat with urease. We coat also with an antibody, which is specific for an overexpressed receptor on the membrane. And then we see how we can break apart this bladder cancer. The first point would be, can they move in urine? This is the first point, where urea is a major content. And then the answer is yes. So you can see here in simulated urine how increased the diffusion coefficient, but also in real urine from mice. So okay, the first component is done. The second is like, let's generate the bladder tumor spheroids. Okay, and this is unpublished, that also was published in this ACS Nano last year. We have this spheroid with the receptor here. And the next point will be, how can we use them? Okay, again, as I said, like we have the antibody coating the particles. We have the spheroid here. If there's no urea in the media, you don't see too much inside, fluorescence inside. There's not too much internalization, whether either if we coat it or not. But if we now have urea in the media, the nanoparticles start to move. They start to go towards much faster to the spheroid, and also because they have the antibody, they can penetrate. So we can see here a huge increase in the tumor spheroid penetration when there's fuel and antibody. But also, it's very important, we can suppress, we can stop about 50% the cell progression, which is very important for stopping the development on evolution of this cancer spheroid. Okay, so all this was done in vitro, of course, and now there are some papers which says that in order to do something in nanomedicine, you need trillions of these nanoparticles. So imagine like having this swarm of fishes. So this is what we need. We don't need one particle moving around, but we need millions of them. And this is something unpublished that we are working on in our lab, and will be a matter of my EST consolidator, I would say, and in the next future probably in the community, how collectively things move. What we are seeing here is a recent video where we have millions of these nanomotors moving around, making a swarm, and I will tell you a little bit about that, what we do with these swarms of nanoparticles. But the other point would be, we need to see them, not only having the swarming, but also see them in vivo. A couple of years ago, together with people from Maune, we reported this work on a much larger microtube, or actually it's a microjet, which we put in the PET CT in some phantoms. And here on the tube one, these are hundreds of thousands of these micromotors. And this is tube one has no fuel, but tube two has fuel. And I think probably you can see in this video, this is time lapse and this is speed up. This is about 15 minutes how this swarm is moving from one side to the other, but still was too large. Okay, and today is about nanoparticles. What is out there? What has been published? So there are a few works reported on using medical imaging from this work from Wang, and then Gianavachi, and then also with Salvador Pané, and then this from Dresden. And also if you go to ex vivo, some other technique OCT by Fisher. But interestingly enough, only 2 works have been reported for in vivo. Well, I think this was also ex vivo, in vivo from Joe, and fluorescence. but using medical imaging and in vivo, there are a couple of works, so we can refer like a bunch of them, and we are very interested. I think this is very timely to go in that direction. So what we do is we take our nanoparticles, we synthesize, not going into detail, this very basic synthesis, but the interesting here is that we, together in the, actually this was done in Maune, they generate the isotopes, and we have two cases, we have fluorine and we have iodine as well, and we can code our nanoparticles, and we can fully characterize them and see that after purifying the suspension, there is nothing supernatant. So every single labeling that you're going to see in the next slides using PET-CT imaging, which is a technique that we think is very, very useful, then we don't have anything in solution. Everything you will see is coming from the particles. Okay. Now, so we have these swarms under the optical microscope, and not only these images you saw before. Now we want to image like the heat map, which indicates the residence time. So how long a nanoparticle is staying in one place. This is what you can see here. On the left side, you have a swarm. These are 100. Again, I don't know how many, can be millions of these nanoparticles, and they all mainly stay in the same time. So the red, more red, will be longer time in the same place. So only flow is here, but here you can see there's a lot of convection flows going everywhere. This is the place where there's lower residence time, so it means there's a lot of moving in solution here. But also one can check, so they do move, but do they move the fluid? This something is known in the community. We also reported a few years ago that micromotors have micromixing. So here only, so we did this particle image. Sorry? Okay. And then we can see here on the right side a huge, like the vectors, showing a huge change in the flow migration. So that means that it's changing, is mixing the solution. Okay, let me go rather quick now. We want to now move to PET-CT. This was all optical. Now in PET-CT, what we're doing is microfluidic channels, and I don't think I have time to go through that, but just to tell you that through this channel they can go and we can image them. And this is a huge quantification that we have done in our lab. The next step will be, can we see them inside the body? Okay, so we put through the tail of the rat, and then we observe in vivo the imaging of these enzyme-powered nanomotors. This is not so crucial because you might remember I talked about bladder. Okay, this is just an idea that, let's say, we don't kill the mice, and we can see that they don't accumulate in the blood in the kidney. They follow the typical progression through the liver, and basically not much more. Here it comes to the interesting story, which is that we inject this directly intravesical through to the bladder. So you might not see too well here, but this is coming from this PET-CT. This is the mice and this is the bladder, and here we have a cloud. Then the reddish part will be our nanoparticles with the isotope, and we identified two regions of interest to see whether this micromixing that I was telling you about is happening or not. Okay, so this is the successful case. Okay, so we have initial time when we inject, and we have after 45 minutes. And we see that after this time, there's, I mean, the two clouds start to separate, but then with time, they merge together. So that means that the whole suspension is homogenized. It means that the nanomotors have moved through the bladder, have gone through all the solution and reached also the walls and reach everywhere. So if you can inject also some drug and mix them together. If you have a nanoparticle which has no enzyme, so there's no migration, we see a phase separation. So this is not good for drug delivery. Okay, so this is an interesting point here. And we have another control. People might say, okay, maybe the nanoparticle before, because it has no protein, no enzyme, is leading to this progression, but no, we have the control. We coded the nanoparticle with BSA, and we observe for the controls and different, like with urea, without urea, we see the same two phases separation. So indeed, there's a good mixing by the nanomotors, but also we can image them, which is very, very novel, I think, and impactful in this field. So I hope in the beginning, this part of my talk, I convinced you that we can have some nanobots that are self-powered. I don't have to do anything. I just put in solution. They take the fuel from here, they make a chemical reaction, and they move from point A to point B. I didn't tell you about today about motion control, but there are a lot of people working on motion control ourselves, but it's more about biocompatibility. We are moving towards biocompatible systems because we want to do biomedical application, but without forgetting very crucial, important fundamental aspects. There are other aspects also on the enzymes, like how to bind the enzyme. We have seen, we are submitting now these days a work like how to bind the enzyme is very, very important for the catalysis. We use them as active drug delivery system, as targeting systems, and also for imaging. And we hope we can use this for theranostic application. Okay, so the last part of my talk will be to go into the big hybrid systems. So this is the last part of this larger scale. And as I said, we want to mimic some larger robotics. And for that, we bioengineering 3D printed actuators. Okay, so here is a bit different. So we went from nanoparticles with enzymes. Now we're having tissues. We have complex system with many cells embedded into gels. And we want to, this is based on skeletal muscle tissues. Okay, there are two types of actuators in the literature. One is based on cardiomyocytes. They are very strong, very powerful, but you cannot control them, and they are hard, and you cannot stop them. They synchronize, they coordinate, but there's no stop on the motion. And also there are skeletal muscle tissues. Skeletal muscles react with electrical stimulation or with light, optogenetics, so you can control the actuation. And we use them as force testing platform. So we can add components, we can do many things with this. We can measure the force of the muscle. We can lead to aging as well. We can tune, and we can even print human cells. And we want to do also big robotics. Okay, so how we do this? The trick is to bioprint. So we have a cocktail that took us really long time to develop this cocktail with C2C12 mouse, myoblasts, fibrinogen, gelatin, hyaluronic acid, and then with thrombin, we lead to the cross-linking, and this is real time as we are printing this bioactuator, this shape that after some days they will mature and then nucleate into myotubes. It was very important, as you see in a muscle, fibers are aligned, and to our surprise, while we were printing, we saw a very nice alignment. You see here the angle is 0 for all the structures, so all are the way you are printing. You can see an alignment of these fibers, which is extremely important for the actuation. Then we apply electrical stimulation, so it means we put 2 electrodes on the sides or at the bottom. You can have flexible sensor, you can have many things with that. And you can see also the calcium, by calcium imaging, you can follow all the peaks, how it's beating. So let's say if we apply 1 Hz frequency, the muscle will react in 1 Hz as well. In order to do an actuator, you need some weight or some post, some measurement alignment. So here what we did is we had printed this ring around 2 pillars. Okay, I will show you later as well this one. And then the muscle starts to actuate, and then we can follow the reference of this pillar, how much is bended. And this again, you apply different stimulation, and 1 Hz, you can see very nicely all the peaks, and even to 10 Hz, and then you can stop it down, and you can play a lot with this. And even more, the more interesting part here, it comes for the exercising. You can actually train your muscle. So remember these 2 pillars. Here we take the bending of the pillar. This is the printed PDMS pillar. And here you will see how over time we are training it. Let me see if we can play the video. No, I don't see the video playing here anyway. The video will show or might show you that with time this pillar will be having a higher displacement because we are training. So it means that you exercise over 2 hours and then it becomes like 2 or 3 times stronger the pillar. Okay, so we can measure if you add drugs or you add any ingredient into the muscle. This is very important. Can you make something, a muscle relax? Can you make a muscle stronger? Can you play with that and overcome the aging, for instance? So nowadays we have two topics in that. One is confidential with a private company we are using for some components, and also we can have some ongoing projects with some collaboration, internal IBEC and external 3D print human cells with different pathologies, which will be very important for biomedical applications. Okay, last couple of minutes of my talk will be about these swimming robots. Okay, so we look at the state of the art, and we saw by Bashir's work in PNAS 2014, and then there were a couple of papers afterwards how to make something that swims. So this is a ring again of skeletal muscle, which is around 2 posts, and this is swimming. Pay attention to this video because you see some symmetric post. So this leg is larger than the other one. So that makes the boat moving in one direction, but also this is speeded up 3 times. Okay. So what we need first is to have a very compliant design, something that bends. This I cannot see it as well. Anyway, so this is bending. So we have the ring putting around this thing, and here you can see how changing the pillars by this bending structure or the spring-like structure by electrical stimulation, we can have a contraction as we want. And this is the schematic, how we fabricate this biobot. Again, myoblast, we make it into a ring, and then we put around the spring, and then this bending will lead to an actuation, as you can see here, but still not moving. We are working a lot on this design. Here, we can make it symmetric. We can make it asymmetric, as the paper I showed you before. And now Rafa is doing this Maria's work, and Rafa is working on the finite element analysis, optimizing what's the best structure for us, what's the best material and the composition. And we observe, just a comparison, we observe that very interesting, if we put the ring around the spring-like structure, which is compliant, means that there is some deformation, but also like spring, it goes back, then the force that we observe by the spring after 14 days of maturation is much higher than if we use the pillars. Again, this for some reason this is not playing. Now everything is playing here. Okay, so you will see here, like we also observe spontaneous contraction, which is not observed by the pillars. We see a much better force and stronger if we have the electrical stimulation on the spring than in here. Okay, are these going to move? Yes, the answer will be yes, because otherwise I will not talk about robots. But for that, we need 2 electrodes. This is our setup. We put the electrodes far distance, and now we start. They start to swim. Okay, so we apply 1 air. You can see here the magnification. Sorry, maybe it's 2. This little guy is almost transparent, so it's a bit difficult to see. Here we make with the code, we follow, you can see the trajectories, time lapse, and you can see how it's jumping by applying the frequencies, making a jump and step-like motion, 1 hertz, 2 hertz, and 5 hertz. We can quantify the motion. And there's no drip here. You can see, like, no matter the position of the electrodes, it will go one way or the other. So this asymmetric biobot is moving at decent speed. And what is this speed? The speed is basically, we compare with the state of the art. We are now normalizing this time, actually going down to real time here, going to real time and the same size. So we are not that bad in speed. Actually, our little guy is slightly faster than the PNS published a while ago. Okay, so I hope I could convince you that we have some small robots. We don't have a finger, we don't have a big one, but this is one step further into making these hybrid robotic systems. Here there are some collaborators. There are many more collaborators, basically in other sections and things that I haven't reported today. Thanks to the NICE team, a very motivated, hardworking guys, and also the funding agency. And thanks for your attention.